introduction, and also thanks to the HFSP to by giving the chance to play on this project with a bunch of very interdisciplinary people, quite crazy because they decided to work on this project that's talk about focus on sex and alcohol. We will see in a bit. Well, everything started from a first observation that Saccharomyces cerevisiae, the budding yeast, can be present in natural environment thanks to the activity vectoring of social wasps. In fact, social wasps are capable of bringing with them in their intestine a quite broad genetic variation of Saccharomyces cerevisiae, all the red dots in this tree here. But another particular characteristic of these strains isolated from this environment is that they present a very high level of heterozygosity. Heterozygosity in Saccharomyces is linked to outbreeding. That is a process that doesn't occur very frequently in natural environment. In fact, Saccharomyces cerevisiae natural strains are mostly diploid. Diploid cells can reproduce themselves over budding, but then if there's a stressor, components of the environment, they can sporulate. Those spores are maintained within a very strong structure, the ascus, that keep them safe from the environment, okay? Until the environment get better, and so the spores can go back to vegetative cell life, and so germinate. And if the ascus is not broken, what happens? They tend to mate with each other, okay? Spores from the same parental strain can mate to each other, reducing or maintaining the heterozygosity, and so this is called inbreeding. In the wasp of gut, it seems to happen the outbreeding, so spores released by the ascus can germinate, meet with spore from other strains, and so increase the heterozygosity. And since this is a very peculiar event to occur in natural setting, we started asking what happens inside the wasp intestine. They must be presenting to the yeast of combination of different environment, situations, stressors, and stuff like this, promoting this process. And first of all, we verified whether this inter-strain mating really occurred in the wasp intestine. And so we fed the wasps with a combination of different strains, diploid and haploid of Saccharomyces cerevisiae, and both after a very long period of hibernation, 3 months of hibernating giants maintenance, but also in active wasps, so very faster, just already after 1 week, we could observe the generation of hybrid, so strain produced by the mating of different strains. So we confirmed that the hybridization, the outcrossing can occur within the gut of the intestine of wasps. Okay, once defined this, we asked, okay, what are the characteristics that promotes this complex pathway? And we started thanks to the activity of the activity of a combination of entomologists, Francesca Barbero, Luca Casacci, and microbiologists, myself and Silvia Above, the University of Turin. We started investigating the response of Saccharomyces cerevisiae to the environment by using molecular analysis. The first one was classical transcriptomic. We fed the wasps with Saccharomyces cerevisiae. After 3 days, we took the RNA sequencing analyzed the data. Another approach was the use of yeast deletion collection. That is a pool of different strains, each one deleted in a single gene, okay? And every single deletion is codified according to the presence of short sequences. So we fed the wasps with this deletion collection. We had a control optimal condition that we use in the lab. After 3 days, we collected the DNA, sequenced it, and so quantified the different strains, okay, according to the tag. Those strains, reducing their abundance in the wasp, were those deleted in gene fundamental or necessary for the survival in that environment, okay? Thanks to this the combination of these analyses, we could observe that in the crop, Saccharomyces cerevisiae needs, or at least are essential for him, some genes involved in mitosis, so to they need to sporulate in this environment. Okay. On the other hand, in the crop, in the gut, the second part of the intestine, transcriptomic analysis revealed that strains in this environment express at a high level genes involved in alcoholic fermentation. Okay, so from the common sugars, they can produce ethanol. Quite harsh for the poor wasp, because ethanol, high level of ethanol is not, it's quite toxic. But we will see in a bit how it works. Okay, now we have identified the response of the molecular response of yeast, and we wanted to understand what's the condition, chemical and physical condition, promoting this environment. And here we have Elizabeth New and her group from the University of Sydney. They are chemists, and they produce specific probes allowing us to quantify some environmental characteristic potentially linked on imposing some response in Saccharomyces cerevisiae, among which pH, viscosity, available nitrogen, and saccharides. Thanks to this set of probes, we could quantify that crop is actually a quite good environment for Saccharomyces cerevisiae to survive. It's similar to the optimal condition that we have in the lab, okay, with neutral pH, a moderate level of viscosity, a bit more than water, but not too high, moderate nitrogen, and low saccharides. On the other hand, the gap is much more stressful for yeast, not much because of the availability of nutrients, because of course that's the place where the wasp have the highest level of digestion of their food. So, but of course they have a lot of nitrogen and saccharides concentration. But actually the very basic pH and high viscosity is something that is not really liked by Saccharomyces cerevisiae. Okay, so we started wondering, how can it survive in this environment? And we tested them, this condition in silico, okay, in vitro, by inoculating Saccharomyces cerevisiae cells in settings characterized by the feature that I just showed you. And we observed that actually in the crop, the red part here, cells are capable to grow as in optimal condition that we use in the lab. But also in the gut, especially this blue here, they are capable of reproducing the cell a little bit late. I mean, it takes longer, but still they are capable of growing. Maybe the fact that it takes longer for them to reproduce is also due to the fact that in the gut we observed a clumping of cells compared to the other situation in which the cells are more capable of growing by themselves. Same goes for the spores. We inoculate the spores in the same condition, and we observed their capability of germinating. In this case, we were very surprised to observe that in the gut, so the harsh condition for Saccharomyces cerevisiae, were more fast to germinate. So eventually, this environment promotes the germination of spores. So if you remember what I just told you about the clumping, this means that in the crop, in the gut, the cells are capable of germinating, the spores are capable of germinating faster, and if they are clumping, they might be more, for them might be easier to mate with a strain from another, a spore from another strain. Okay, all these information were fundamental to fill in, let's say, to test the results in a dynamic flux balance analysis performed thanks to the model comets prepared by Daniel Segre of the University of Boston. And this was, the model was used in two different approaches. The first one, this one, consisted in testing the ability of the Saccharomyces cerevisiae cells to grow in the absence of one gene at a time, like we did with the experiments, the deletion collection I told you at the beginning. We did the same in with comets, okay? And we've evaluated the maximal growth ability of these settings. both using as nutrients, so environmental settings, the crop, and the gut condition that we have quantified previously. And again, we had good results because there was a confirmation of the genes that we observed by means of experimental data, the deletion collection analysis, and most of the genes were confirmed to be essential also in this case. And again, we had highlighted the needs of alcoholic biosynthesis processes. So we confirm also the results that we had by means of transcriptomic analysis. The second approach we used this model was to predict the capability of growing of the single cell, so producing a growth curve, and also at the same time quantifying the usage of metabolites and production of other metabolites. This again in the two settings, crop and gut conditions. In addition, we also tested the presence of aerobic or anaerobic condition. And actually, what we observe by transcriptomics resemble exactly what we notice by using the model in anaerobic condition. So basically, a slow grower grow in the gut condition and production of ethanol. Again, ethanol, toxic stuff. But then, luckily, last year there was a publication, a study that showed that actually social wasps are really the best animals capable of tolerating ethanol, also because they can metabolize and use them as carbon sources. So this might also explain why the association between Saccharomyces cerevisiae and social wasps is so strict. Okay, so just to sum up, what we observe is that in the crop of the wasp intestine, Saccharomyces cerevisiae is capable of sporulating, but then moving to the gut, the spores are capable of germinating very fast, and potentially it can also mate with each other thanks to their clamping. What we miss so far is this part here. So I told you at the beginning that the ascus is very strong. Okay, it needs to be broken to promote the mating between different spores of the of different strains. Okay, so apply. Then was introduced Marco Polin, that is a physician of the group from the University of Balearic Islands, that started to predict, to evaluate whether the strength of the muscles, of the intestinal muscle, was enough to break physically this ascus. Unfortunately, it wasn't. It needs a lot of more strong strength, okay? So we started to investigate the potential presence of enzyme, either from the microbiota, the rest of the microbiota, or even produced by the host. And first of all, what we did was to evaluate the capability of how the ascus behave when exposed to the crop or to the gut content compared to the enzyme that we commonly use in the lab, enzymolyase. It's the best enzyme to break the ascus. And what we observed is that actually crop and gut content are capable, at least as much as the enzymolyase enzyme, to break the ascus. Interesting. Unfortunately, social wasps are not very well defined, not in terms of genetics and physiology, so we started to investigate why could it be. Could it be something produced by the host or maybe by the other microbiota? Actually, we have, it's available, the genome sequence of the wasps, and at the same time we performed transcriptomic analysis of the wasp in response to the yeast. Luckily, these two kind of information pointed out to a specific protein that was both highly expressed in the presence of the yeast, and also showed a domain exactly identical to the one that is present in enzymolyase, the enzyme that we use in the lab. And by performing a prediction of the structure of this enzyme, potential enzyme, we noticed that the substrate of enzymolyase, beta-glucan, perfectly fit within the domain of activity of this enzyme. So maybe we have the completion of the entire pathway towards outbreeding. Still working on it, but stay tuned. With this, that's all. I would like to thank again all the people that contributed to this project, HFSP, and you for your attention.